Thank you for joining us today. I am Joel Shepard, President and owner of Shepard Specialty Papers. The talk is being presented by Dr. Andy Schile from the Jackson Laboratories. It will describe how the Jackson Laboratories has used cage enrichment to improve challenging situations with various mouse strains, including barbering in B6 mice and aggression in highly diverse JDO outbred mice. He'll also describe enrichment strategies to improve breeding performance and pup well-being, as well as share their experiences maintaining large colonies of aging mice. Dr. Andy Schile provides scientific oversight for their largest mouse colonies. He specializes in strain characterization studies and housing and husbandry projects. Some recent projects have focused on understanding how aging mice can serve as improved disease models. He has a PhD in biology from Rockefeller University and has worked at JAX in Sacramento since 2008. With that, I'm going to give the floor to Dr. Andy Schile.
Great. Thanks very much, Joel. And it's a real pleasure and a privilege to be here today. I'd like to thank Joel and Shepard Specialty Papers for giving me the opportunity to speak to you about some recent projects that we've done at the Jackson Laboratory, many of which feature enrichment devices that are manufactured by Shepard Specialty Papers. And we'd like to share with you some of our experiences over the last few years with some recent studies that we've done. So some of these findings have been presented at the national ALAS meetings and regional ALAS meetings as well. But I hope that this is a very helpful kind of overview and summary of enrichment studies that we've done with a variety of different strains. So you probably know the Jackson Laboratory as mainly as a distributor of laboratory mice. So we distribute over 11,000 different strains of laboratory mice to the research community. But there are other important parts to our nonprofit mission that I'd like to point out as well. So in addition to distributing mice, we also provide a variety of different resources some of you may be familiar with. A lot of these resources are accessible at no charge via our website, so related to bioinformatics and information about mouse strains, et cetera. But another key part of our mission, and a big reason why we're here today, relates to educating scientists. So this ranges everything from in-person conferences that we will start offering again when the time is right, but principally a lot of our education efforts have focused on online learning these days. So the benefits of enrichment for laboratory mice, hopefully by this point, don't need any introduction in the community. By this point, it's very clear that enriching the cage environment encourages natural instincts that mice have to produce or seek shelter, burrowing instincts, things like this. And importantly, this provides them with a degree of control over their environment in the cage setting. There are data showing that enriched cage environments lead to improved brain and neuronal development, and they can also lead to reductions in kind of undesirable aspects or behaviors that we see in laboratory animals, which could include things like stereotypies and barbering. And that'll be a big focus of today's talk is how we can use these different types of enrichment to try to reduce some of these undesirable traits and improve well-being of the mice overall. The first study that I'd like to talk to you about was one that was presented at the national ALAS meeting 2 years ago, I believe it was. And that is a very large enrichment breeding study. So this was a breeding study that involved the C57 Black 6J inbred strain, which many of you may know. This strain is the kind of the most commonly used variety of inbred laboratory mouse in research today. And so the C57 Black 6J strain will feature prominently in a lot of the experiments that we talk about today, simply because it is such a commonly used mouse strain that has a variety of its own kind of issues that have to be contended with that I'll talk with you more about later. But this project focused specifically on enriching the cage environment for breeders. So what we did is we established 5 different groups in our study, and each of the different groups received a different type of enrichment in their cage. So I should point out that all of the cages had enrichment in the form of the bedding that we provide them. So all of the mice at the Jackson Laboratory in our production breeding facilities and elsewhere receive a a blend. It's roughly a 50-50 mixture of aspen chips and aspen shavings. So you can see the aspen shavings here, and these serve as a type of nesting material that's just naturally in their bedding. But in addition to all of the groups receiving aspen shavings in their bedding, there was another group that had the aspen shavings plus a cardboard hut. So this is the shepherd shack. It's a cotton square. Another group got twisted paper enrichment. And then the fifth group got a cardboard tunnel, right? So these are all autoclavable forms of cage enrichment. And so they all received the same husbandry procedures, the same diet, water, and the bedding mixture. But really the difference here was the type of supplemental enrichment that was provided to them. And another thing we did is that we rotated the technicians among the different groups to try to ensure some consistency in the data collection, and just to make sure that there weren't perceptions, individuals' perceptions that were maybe contributing to some of the different findings that we saw here. So I'll tell you about the parameters that we looked at for each of the cage in the study. So this was a breeding study, so we were interested in basically two major categories of data. So one is related to productivity, but another one very key is the well-being of the animals that were produced in these environments. So we're recording the number of litters, how many pups were born and weaned, the percentage of those pups born that survived to weaning, mortalities that were seen among the pups, and any animals that had to be euthanized for abnormalities at the time of weaning. So these would include things like the number of runts that we saw and the number of pups that were showing signs of barbering, so hair loss or whisker picking. We're also interested in knowing how many of the cages experienced flooding. So at the Jackson Laboratory, we provide water in bottles. And so any watering system can develop some leaks, whether it's automated or water bottles. But we were interested to know if any of these enrichments could reduce the number of times when we find that a water bottle has leaked into the cage. As you can see, this was a very large study. Each one of our treatment groups had approximately 600 different breeding trios in them. So it's a very well-powered study. And this is just the number in parentheses is indicating the percentage of these cages that survived to the end of their 30-week breeding period as an intact productive trio. So overall, these numbers were quite good. So let me tell you about some of the data that we saw. Overall, the type of enrichment had pretty marginal effects on the litter counts. So if we just look at the number of litters that are born for a breeding trio, and I've kind of ranked them here going left to right from smallest to largest, cotton squares having the lowest number of overall litters per breeding trio, with the tunnel group having the largest number of litters. What you can see though is that there are some significant differences between the groups, but the magnitude of those differences are really quite small. If you look at what the y-axis is showing us, so the number of litters per breeding trio, is that the difference between the group that had the fewest litters, which 9.3 on average across the entire breeding period, versus the one that had the most of 9.8, is a fraction of a litter. So obviously mice aren't going to have a fraction of a litter. So we would say that there were significant effects in the number of litters that there were, depending on the enrichment group. But overall, it was kind of a marginal effect that we saw here. So the group that had the most number of litters here, we're looking at the group that had a cardboard tunnel in their breeding box. So here's a graph that's actually showing 3 pieces of data. So let me walk you through that. So we've got these bar graphs that are indicating so the number of pups that were, say, for example, either born as shown in the black graph or weaned as shown in the blue graph. So these two graphs are overlaid on each other, meaning that they share a common axis here. So in the cotton square group, for example, a breeder in this group on average would wean 42 out of the approximately 56 pups that were born throughout its entire breeding period. And on average, that works out to about a 75% average survival to weaning for this group. And so what we found was that the type of cage enrichment that was provided to the breeder could have a significant effect on a variety of different things that we looked at here. Where, for example, if we look at the number of pups born, I'm indicating which differences, pairwise differences were significant over here. Where, for example, the difference between the number born to the cotton square group versus the twisted paper, or cotton square versus hut or tunnel, that this difference here was significant. So you have significantly more pups being born in these three groups compared in a pairwise manner to the cotton square group. We saw significant effects on the percentage of pups or the number of pups that survived to weaning, as well as the percentage for each individual cage, where the groups that had the highest percentage of pups surviving to weaning were either the tunnel group or right behind it, the twisted paper group. So these two enrichment devices, when sort of used in combination with the enrichment that's naturally going to be present in the bedding mixture that we use too in the form of aspen chips, had a significant increase in the percentage of pups that were surviving to weaning. So when we looked at the reasons of why different enrichment materials offered benefits in this breeding study, we found that if we look at the number of mortalities, so these would be pups that were found dead or went missing and presumed cannibalized, we found that the breeding cages that received a twisted paper enrichment, so the alpha twist, had the overall lowest numbers of mortalities per cage. So it's between 2 to 3 mortalities throughout the course of the entire 30-week breeding period, where this group, the twisted paper group, was significantly fewer mortalities than any of the other enrichment groups that were found. So the provision of the aspen chip or the alpha twist in the cage in the form of the twisted paper led to the fewest number of mortalities. So this percentage here that's shown, so 4%, indicates the total number of pups born on average during that 30-week period that experienced a mortality. So it would be 4% here, which was a significant reduction over the other groups. Okay, if we look at the reasons why we had to, or if we look at the abnormalities that pups that we saw in the pups that required euthanasia, we found that the number of the pups that had to be removed for abnormalities was minimized in the groups that had a tunnel. So we have the twisted paper leading to the fewest mortalities or pups being found dead, and the tunnel group had the fewest pups that had to be euthanized for abnormalities. So these differences were significant between the 4.5% of pups born requiring euthanasia for an abnormality versus some of the other percentages that we're seeing here and the overall numbers. So we found that the tunnel group led to the fewest number of pups that had to be euthanized for abnormalities. When we look at what those abnormalities were, the major factors that we're looking at in the C57 Black 6J strain are runts and barbered pups, where, for example, we're showing the number of pups that had to be removed at the time of weaning for being a runt, so abnormally small, and versus the percentage that had to be removed for excessive barbering, which is probably caused by the parents. We found that the tunnel group minimized the number of pups that had to be removed for runts, and also minimized the number that had to be removed as barbered animals. So what we found is that the tunnel group led to reductions in the two most prominent categories of reasons why C57 Black 6J pups need to be removed for an abnormality. So we have the reduction in mortality for the pups that were the groups that were provided with the twisted paper, or the alpha twist, and then those that had the tunnels had fewer abnormalities that required removal at the time of weaning. We found that the tunnel group had the fewest wet cages. So this is telling you of all the cages in our study, how many of them experienced a leaky bottle. So it may have something to do with where the tunnel is perhaps placed in the cage, the type of nesting material that may promote wicking in some instances. But we found that overall, if there was a tunnel in the breeding cage, that we had significantly fewer instances when a water bottle was found to leak into the cage. So there's an advantage in that aspect also. So just to wrap up this quick discussion here about the breeder enrichment project with the C57 Black 6J strain, we found, perhaps not surprisingly, but that environmental enrichment could improve the well-being of pups that are in a breeding colony, and can also lead to increases in productivity, because anytime you have more quality animals being produced, you have overall greater productivity, which of course is important anytime that you're thinking about breeding. We found that enrichment trials and comparisons can help to determine the best type for each strain, and that's a theme that we're going to come back to throughout today's presentation. And that we're also very interested in seeing about some additional enrichment combinations to try to further reduce the number of losses that we see among pups and sort of look at the overall quality of the animals being produced. So I'd like to talk to you now about some other strain-specific challenges that we have approached using enrichment. So I mentioned the C57 Black 6J strain in the breeding study, but you probably know this is the most commonly used inbred strain in research, but it's prone to a variety of abnormalities that it can include eye defects, hair loss, hydrocephalus, dermatitis. So these are issues that are prevalent in the C57 Black 6J strain to some degree. Another strain that we wanted to investigate was the SJL strain. So this is a mouse model. You may be familiar with this. It's very commonly used to make F1 hybrid mice for a variety of projects, but it's also used as a model for studying multiple sclerosis. The major challenge with this strain is that it's aggressive. The SJLJ males in particular are among the more aggressive male mice that we work with at the Jackson Laboratory. So we wanted to see if different types of enrichment could try to improve some of these challenges that we have with these two different strains. So the type of barbering that occurs in C57 Black 6J mice has a female bias. It'll commonly present with trimmed or picked whiskers and hair loss, certain type of alopecia. This is known to be affected by age, so older mice are more commonly affected, and also cage density. And there are a variety of explanations that have been put forward that are beyond the scope of today's presentation that try to explain, why these conditions develop in the C57 Black 6J strain. But it's definitely an issue that we and others who work with this strain must contend with. And we wanted to see if we could use enrichments of the cage environment to try to reduce the incidence of these conditions in this strain. So one of the approaches that's been used in the literature is what's called a rotational enrichment scheme, where every week these groups of same-sex group-housed mice are given a new type of enrichment. So we designed an experiment where for 1 week, the mice would get gnawing enrichment. So this would be something like a gumma-bone, a nylabone, and a wooden gnawing block. The next week, at the time of cage change, they were given these metal trapeze and shower curtain rings that could hang from the wire bar lid that's present in all of our cages, kind of where the wire bar lid, of course, is where we put the food and the water bottles. The next week, they would get a cardboard tube and a shack. And so we would just change this on a weekly basis. So they'd get a new enrichment every week, or a new type of enrichment every week. We did this in kind of a limited experiment where we had 2 sets of 120 female C57 Black 6J mice that were assigned to one of 2 enrichment groups. So one of them got the rotating enrichment, and the other got no extra enrichment. They still had enrichment in their cages in the form of the aspen shavings that provided nesting material, but they didn't get the supplemental rotation of the enrichment groups. And we followed our typical husbandry practices and cage changes, et cetera, over the course of 18 weeks. And what we found during the course of this 18-week study is if we just look at the percentage of mice in the study that were showing signs of hair loss, so this is happening in a group house situation, we found that, so the group that had rotating enrichment is this red line, is that over time, over the weeks, we didn't start to see signs of hair loss in these mice until they were 14 weeks of age, or 14 weeks on study, excuse me. But the group that didn't have any extra enrichment, so that's shown in this black line, you can see that you start seeing signs of this, the hair loss or the barbering that happens earlier and to a greater extent. So by providing this rotational enrichment scheme, we were able to reduce and delay the incidence of the hair loss that was happening in the female mice. So some of the obvious benefits of this are the novelty and diversity provides, and it was effective at reducing the hair loss in these mice. But the challenges, of course, are the cost. You need to have all these enrichment devices, and there are also operational challenges in making sure that you have enough on hand in the mouse rooms at all times to be able to rotate this, and you would need to have a set schedule for this week, these cages got this enrichment, and so on. So we wanted to see if we could understand if there was any one of these groups that would offer the same offer the same benefits as the rotational enrichment. So what we did is we did a larger study where instead of rotating the enrichment form that was used, we created 3 different groups that got the same enrichment throughout the entire study. So one group got no extra cage enrichment beyond the aspen shavings that are present in their bedding, of course. The second group got a cardboard tunnel and a shack. And the third group got trapeze and rings in their cages. We didn't create a group with the gnawing enrichment because we found that in our initial study that the C57 Black 6J females at least tended to ignore and even in many cases bury the gnawing type enrichments. We didn't really see much evidence that they were utilizing these for their intended purpose. So what we did is we monitored mice between the ages of 3 and 12 weeks for hair loss and other adverse signs in the populations. So when we compared shelter and activity type enrichment in the C57 Black 6J females, we found that the group that had no extra enrichment, so this is their age in weeks on the bottom, would have the appearance, start to see the appearance of mice with hair loss about 7 weeks of age. We found that we would see evidence of this in the group that had the activity enrichment in the form of the trapeze and the rings on their wire bar lids would increase. But the groups of female mice or the cages of female mice that had a cardboard tunnel and a shack or a dome had pretty minimal, were minimally affected by this compared to the other types. So we found, at least in this strain, that the shelter type enrichment offered many of the same benefits that a rotational enrichment did, with the advantage of a consistent product that's added at all times to the group. So I'll come back to what our experiences were when we rolled that out on a bigger scale in a moment. We also wanted to know if different nesting materials can reduce the aggressive tendencies that we see in SJLJ male mice. So the type of aggression that we see in this, the Jackson substrain of the SJL strain has a male bias. So often what will happen is that the male mouse in a breeding cage can attack his mate. If he's present with pups, he can attack them. They will also fight with each other in same-sex group housing, so cage mates, whether they're litter mates or not. And then generally speaking, the aggressive tendencies do tend to increase with the age and the cage density in this particular strain. So in this study, we set out with a slightly different study design to look at, is there a particular type of nesting material that can try to reduce that aggressive tendency in the SJLJ strain? So this study design was a little bit different in the sense that all of the study groups here, this is in a breeding study, all of the groups received an aspen chip bedding, but then they varied in the type of nesting material that was present. So in the first study that I discussed with you, all of the cages, the different enrichment groups had a consistent presence of aspen shavings in their cages. In this group, they didn't all necessarily have aspen shavings, rather they all had aspen chips as the absorbent bedding material. But then in addition to that, they would either have the twisted paper, so the alpha twist, the aspen shavings, crinkled paper, or a cotton square. And so what we did is we monitored the productivity and different welfare/well-being parameters throughout a standard breeding period for this strain, which in this case is 22 weeks. It's shorter than the C57 Black 6J that I talked about earlier. We also followed our standard husbandry practices for this strain. So we did find that there were a few significant effects of the type of nesting material on some of the different parameters that we looked at. So going left to right, this is looking at the number of pups surviving to weaning on a per breeder basis. And we found that the number of pups that survived to weaning was maximized in the group that was provided twisted paper as their nesting material, so the alpha twist. The fewest number of pups that survived to weaning were in the group that had just the aspen shavings in their group, so aspen chip and aspen shavings in their cages, excuse me, and the cotton square. So these were the nesting materials that led overall to the fewest number of pups surviving to weaning. We found, though, that the benefits were probably related to the number, more related to the number of litters that were produced overall. So if we look at the average number of litters that were produced per female in the different groups, we found that the number of litters per female was was most in the group that had the twisted paper or alpha twist in their bedding, and it was kind of lowest in the aspen shavings and the cotton square group. So the number of pups that are surviving to weaning are very likely related to the overall greater number of litters that we're seeing in the group provided with the twisted paper. And we found a productivity advantage in the sense that cages, SJL breeding cages that had the twisted paper in there, had the shortest average time to their first litter being born. So after mating the mice, we would find that, that first litter was born about, 26 days later, but it might be longer in the group that had the cotton square. So we were kind of surprised to find that many factors didn't depend on nesting material. So in this particular study, we didn't find any significant effect of the nesting material on mortality, which of course is a challenge in this strain with the aggressive male mice, or the number of pups that were removed for different abnormalities like bite wounds or runting, et cetera, hair loss, pup mortality. Now, none of these parameters that we thought might be affected by the type of nesting material, none of them in this study was significantly affected by them. So the advantages that we found were mostly on the productivity side and not necessarily as having a significant effect on kind of reducing some of those aggressive tendencies in this strain. So we found some different benefits that weren't necessarily what we were expecting, but that's what we found in this study. So thinking specifically about these two examples, we found that hair loss was lessened when female mice were provided extra enrichment. So this is perhaps not surprising. It's known in C57 Black 6J and other strains that providing a more enriched cage environment can lead to reductions in kind of abnormal activities or behaviors, undesirable aspects of the mice in a group house situation. And that we were pleased to find that a shelter type enrichment in this strain offered a very similar benefit as the rotating enrichment. So a more consistent type of cage environment, or a more consistent type of enrichment offered very similar benefits. In the SJLJ study, we found that the twisted paper, or the alpha twist, greatly improved the productivity of this strain, but it didn't have an obvious effect on the pup survival. So for this reason, we've been including that alpha twist, or the twisted paper, in the breeding cages for the SJL, largely because it seems to have a benefit on the overall number of litters that are produced, even though it's not leading to any significant differences in the survival of animals, the survival of pups in the breeding colonies. So I'd like to talk to you about group housed animals, because I realize that not everybody here does breeding of laboratory mice. And in some cases, you may simply be receiving cohorts of mice, either males or females or both, and then keeping them in a group house situation while experiments are ongoing. And so the common way that we experience group housed animals at the Jackson Laboratory, at least in the part of the Jackson Laboratory that deals with breeding and distribution of mice, this commonly comes in the form of mice that we're housing in essentially an inventory. So these would be mice that we have bred, and we want to distribute them to researchers. So when we have these group housed animals, these are mice that after they've been weaned, so they're in same-sex cohorts. Common reasons why we lose mice, or have to remove mice rather, from our group housed situations are instances of barbering, which could come in the form of hair loss or whisker picking, whisker trimming. Aggression can be a reason. So mice that have bite wounds are euthanized. Eye defects, so in the C57 Black 6J strain in particular, anybody who works with this strain knows that this strain is prone to a variety of different eye defects. So mice can get opaque eyes, they can have small eyes, missing eyes, etc. And another, particularly a C57 Black 6J issue, is malocclusion. So the misalignment of the teeth is principally a Black 6 strain issue, but can be seen in other strains as well. So I mentioned to you earlier the study where we showed that if you gave female C57 Black 6J mice a cardboard tube and a cardboard shack, that we had reductions in the number of female mice that were affected by hair loss and whisker picking, barbering overall. So on the basis of that study, we rolled that practice of adding the shelter enrichment out more broadly across many different colonies of C57 black 6J mice. And we found that when we implemented these cages, when we implemented this type of enrichment in the cages at the end of 2013, that the percentage of female mice that were affected by hair loss, barbering, and whisker picking in a group housed situation, so this is not mice being used for breeding, just group housed same sex, is that there was a pretty sharp drop and significant drop in the percentage of female mice that were affected by this. And so here's a picture of a cage. It's a larger cage, a weaning cage that's going to have that shepherd shack or half of the double dome and the cardboard tube in here. So we had essentially a 50% decrease in loss for the C57 black 6J strain. When we rolled this out for the C57 black 6J males, we were curious to see if we could get a similar effect using just one type of enrichment, shelter enrichment. So instead of adding the cardboard shack and the tunnel, we added just the tunnel. And then we found that just adding the tunnel to group housed male mice led to approximately a 50% decrease in the number of males that were affected with the hair loss, barbering, whisker picking, et cetera. So we found that C57 black 6J strain in particular really seems to benefit from using the shelter type enrichment. There's another mouse strain that we work with that we have to contend with the issues of hair loss and whisker picking in, and that's an immunodeficient strain called the NSG or nod-skid gamma mouse. And so what we wanted to do is to see if we could use shelter type enrichment to reduce the incidence of females that are housed in group settings that are affected by barbering, hair loss, and whisker picking. And we found that by adding a double shack, so both parts of the shepherd shack that come attached, by adding that intact double shack to the inventory cages or the weaning or group house caging, that we could see a reduction in the number of females that we were removing from the colony or euthanizing because they were showing signs of the whisker picking, barbering, et cetera. So we found that the presence of the shelter type enrichment benefited another strain, this immunodeficient NSG mouse. If we think more broadly about the losses that we see in breeding colonies, so not just the C57 black 6J, but in other strains, common reasons for pre-wean losses, we're talking specifically about pups, would include reasons like runting, missing mice, cannibalized mice, and also barbering, which is very likely caused by the parents towards the pups. So we wanted to see what would happen if we started adding some of these shelter type enrichments to other breeding strains. So in this case, what we were working with was the BALB-CJ strain. So this is one of the different BALB-C substrains that we distribute at the Jackson Laboratory. And so we struggle with a relatively high percentage of pre-wean losses in this strain that largely improved to some extent when we started providing the breeders with the shepherd shack. So we provide them, the breeders are provided with 1/2 of what's seen here, because this of course tears apart in the middle. And we found that there was a loss in, there was a decrease in the number of pups that were lost pre-weaning in the BALB-CJ breeding colony when we started giving them the shelter type enrichment. Another strain that's very challenging that we experience challenges with is the C57 black calyx inbred strain. This is kind of a distant relative of the C57 black 6 strain, and that we were contending with a very high pre-wean loss percentage of pups. So we weren't seeing as many pups surviving to weaning as we would like. So we sought to address this by providing enrichment in the form of a cardboard tunnel. And we found that in the time before we started adding the tunnel, we were losing approximately 24% of the pups prior to weaning. But then this number went down to 15% when we started adding the tunnel enrichment. So again, this is another example showing the benefits, at least in our experience, of adding a shelter type enrichment to breeding cages. I mentioned earlier the study with the SJL mice. where we found that there were productivity advantages to adding the twisted paper or the alpha twist to the breeding cages. And so once we rolled that out on a much larger scale beyond just the scope of the smaller project that we were doing, we found that the litter sizes increased modestly, but to an important degree when we started adding the twisted paper alpha twist to the cage environment. We don't always find benefits with all the enrichment trials that we do. So for example, here was a strain where this is a mouse that's used for cardiac disease research. It's an atherosclerosis model that has very high cholesterol. And so this was a C57 Black 6 strain background. So it's a Black 6 mutant strain that we were struggling with a comparatively high percentage of pre-wean losses of pups in our breeding colonies. And we tried adding some cotton squares of supplemental nesting enrichment to the cages, and we found that there weren't any benefits to adding this. In fact, we saw that the condition actually got a little bit worse. So, we've got further projects underway to try to find that most ideal breeding enrichment for this particular strain. Generally speaking, in our experience, the shelter type enrichment seemed to help for C57 Black 6J inbred and also mutant strains that might share this genetic background. I'd like to talk with you now about a type of mouse strain that maybe you're not familiar with, but you might be hearing more about in the coming years, because it's kind of a novel variety of laboratory mice. It's called the diversity outbred population. And so this is a population of outbred mice. So outbred mice, of course, are the opposite of inbred mice, where in an outbred population, you expect every mouse to be different from all the other mice in their litters or in their cages. And that's because they're bred in a way to maximize genetic diversity, whereas inbred mice are bred in a way to minimize their genetic diversity, to create a more homogeneous population of mice. So this is a mouse strain that was developed by a very orderly breeding process that involved introducing genetic contributions from 8 different diverse inbred strains of laboratory mice. So this is a mouse family tree that shows the interrelatedness of a lot of mouse strains, some of which you're familiar with, like the C57 Black 6J, and many of which you may never have heard of before. But essentially, this population of mice, this outbred population, was produced by picking 8 genetically diverse strains of inbred laboratory mice. And through a very sort of complex breeding scheme, these animals were mated together in a way to try to maximize the contributions from all 8 of these inbred mice. So if you look at their genetic makeup, they're not all exactly the same like a C57 Black 6J would be. But what we found is that their genetic makeup in a population has about 1/8 contribution from each of the 8 different founder lines. So it's a way to try to create a population of mice that's genetically diverse. Because if you think about it, if you have a really genetically diverse population, then you can use that to try to understand, variation in responses to a treatment in a group of people, or a group of mice rather. That's maybe more relevant to thinking about human populations, in the sense that an inbred mouse strain is great at minimizing the diversity or variation that's due to genetics. But in some circumstances, if you're trying to understand from maybe a population's genetic standpoint, you might want to have a more broadly genetically diverse population to understand outliers in response to treatment. And then you can try to figure out, because we know a lot about the genetics, what are some of the genetic factors that contribute to that. But one of the challenges with this mouse strain is because it is such a genetically diverse strain that's diverse in virtually every way in terms of their size, their color, their behavior, their appearance, etc., is that this happens to be a very aggressive population of mice, especially in males. It can make group housing these animals extraordinarily difficult because there is strong tendencies of aggression towards group housed animals, principally in the males. So we undertook a study to try to identify conditions that would reduce the aggression of these male mice in a group house setting. So if you think about it, some of these studies involving these outbred stocks of mice, you might need to age the mice for a while till they reach an age when you might want to begin some sort of a treatment or some kind of an intervention. But if you have all the mice singly housed because they have aggressive tendencies, then that can be a challenge for a variety of reasons. Especially if you think about a long-term experiment where, mice are social creatures and, singly housing them for a long period of time is perhaps less ideal than a group house situation in many different respects. So we wanted to see if we could try to minimize these aggressive tendencies. We looked at two different types of caging. So this is a disposable cage that's kind of on the typical kind of shoebox style cage that's in use in many breeding facilities or mouse facilities. And then we also used a larger wean box, which we use at the Jackson Laboratory. It's bigger than a shoebox, standard shoebox size cage. It's about 50% bigger. So it's a kind of a larger cage. And we had different bedding or enrichment groups where we had the aspen chips and shavings and a cardboard tunnel in the disposable cage. We had a cotton pad. So this is essentially a very large form of the cotton squares. So it's the alpha pad. And then we had other combinations in the wean box. And we also tried a cardboard divider, which partitions the cage up into smaller sub areas that the mice might have separate kind of nesting or locations that they can be present in the cage. So we had 4 mice per cage and 5 cages. And we wanted to see what would happen in this instances. Could any of these groups lead to the longest survival reductions in aggressive tendencies? So I'm showing the survival over time in the wean cages and also in the disposable cages. So we had to discontinue our disposable cage study because this population of mice, we had some evidence that they were starting to figure out how to chew out of their disposable cages. So we discontinued that. And we continued our study in the wean cages. So these are polysulfone cages. And then what we found was that the mice that were provided with the aspen chips and shavings, so they got a nesting enrichment and a cardboard tunnel, survived the longest. It's still not perfect. There's still a lot of work to be done to be able to define the conditions that allow these mice to be housed in a group house setting for longer. But compared to the other enrichment groups, survival was improved when we had the nesting enrichment as well as the shelter enrichment in this group house setting. So in this case, we were able to see, survival of, 90% of the mice through approximately 5 months of age, whereas the other groups were experiencing losses that were due to aggressive tendencies as reflected in bite wounds. So we were able to show that in this genetically diverse populations that we could kind of reduce some of these aggressive tendencies. There's clearly more follow-on work to be done, but I think this could be helpful in situations where I think going forward that we who work in sort of the laboratory animal field will start seeing more outbred populations of mice becoming parts of experiments to try to understand more about genetic diversity. And so this could be helpful information as we think about group housing them. So if you think about the costs versus benefits of enrichment, there are certain costs, but they're really more financial costs if you think about it, or time. So they relate to training, operations of having all the enrichment devices, the actual cost of the enrichment device, et cetera. But the benefits are obvious when you see the improvements principally in the well-being and welfare of the animals when they're in the more enriched environment. So of course there's a trade-off in all of these cases. And so the key is trying to understand what are the best enrichment devices to use depending on the strain that you're looking at. So we found that enrichment improves mouse welfare and breeding productivity. The different types of enrichment that you use will depend on the strain, which is why we like to use enrichment trials and comparisons to help to determine the best type for each particular strain. So I'd like to conclude in my last 5 or 10 minutes or so by sharing some experiences that we've had maintaining large colonies of aging mice. So we have a program that we've had in place over the last few years where we set aside large numbers of mice every week at weaning that are destined to age up. So I think that those of you who work in animal facilities will probably, in addition to seeing more outbred mice, probably see more aging animals, because as we know, age is a major risk factor for most diseases and causes of death in people. And so having mouse populations that you can use to understand that risk factor will be, I think, increasingly important over the, as the research progresses. So we aged the C57 Black 6J strain up to 90 weeks, which is 22 months of age. In case you're curious, what does that work out to, mouse years versus people years? This is a life stage equivalency graph that was made that sort of shows roughly what is considered a mature adult C57 Black 6J mouse, what's middle-aged and what's old. So you can see that by a mouse is about 12 months of age, it's considered middle age. It's roughly equivalent to a person in their 40s. But then as they get out to some of those older ages, you start to approach kind of what would be considered an elderly age in the human population. And so in the Black 6J strain, at least, some work done at the Jackson laboratory showed that there's survival curves that you can define for different inbred strains, and that at least in males, we expect a very, very high survival rate, and also in females to a similar extent out through this range of ages that we keep in our aging colony. So it was very important for us to have a good sense for what conditions would be tolerated and which would not be tolerated in our aging colony. Because anybody who's ever worked with an older mouse knows that they quite honestly will look quite different from a younger mouse. So what we tolerate in our aging colony are some things that we wouldn't necessarily tolerate in our groups of younger mice. So for example, in an aged mouse, we tolerate more variation in weights within a cage, so within a group of group housed mice. Coat color changes. You can sometimes see the fur moving away from that pure, black, roughly, black coat color that you expect in the C57 Black 6J strain. You can see some graying, for example, that happens. And we also permit mice that have hair loss to some extent in our aging colonies. But in our aging colonies, we don't tolerate a variety of conditions such as the emergence of ulcerative dermatitis, presence of bite wounds, malocclusion. Males are prone to inguinal hernias and other adverse clinical conditions. So if a mouse presents with one of these clinical signs or adverse signs, we do remove it and euthanize it from the aging colony. But we tolerate more of the things on the left that we don't typically tolerate in a colony of, say, for example, younger mice or mice of different strains. So these are mice that receive a standard chow diet, 6% fat by weight. They get filtered, acidified, autoclaved water in bottles. They're changed weekly in ventilated cages, which the majority of our mouse rooms are have ventilated cages at the Jackson laboratory. Or if it's a non-ventilated cage or a conventional cage, they're changed twice weekly. Their bedding and enrichment, we think, is key to their overall survival and success in our aging colony, because we provide the nesting material in the form of the aspen shavings and the extra enrichment in the form of the cardboard shelters that we discussed earlier. So in our experience, we find that through this age range of up to 90 weeks, that few mice are lost on a typical week. So if I look at all of the mice that are present in our aging colony at these different ranges, and I look at the reasons that we either lose or have to remove a mouse from the aging colony because it's developed some adverse clinical sign or phenotype, we find that in this range of 6 months to a year, on a typical week, less than 0.2% of females are lost, a little bit fewer in the males. These numbers start to creep up over time with age, but overall, the loss percentages are quite low in these aging colonies on a typical week. Some of the common reasons why we lose mice in our aging colonies. So these could be helpful for you if you are working with aging mice, to know that female mice, as they start to get older, so in the 6 month to 1 year time point, about half of this 0.18% is due to the presence of dermatitis, so ulcerative dermatitis. Of course, there's sort of an appreciation in recent years that dermatitis can be treated in some circumstances, through the use of toenail trimming. But so far, we haven't been treating these, at least in the aging colonies. Fortunately, the incidence of this condition is overall quite low. And so we're continuing to remove affected animals that have this condition. And we actually see more bite wounds in the females at the 6 month to a year time point than we do for males. So about half of that 0.18% is due to dermatitis losses, and about 20% is due to bite wounds. So as they get older, we still see the presence of dermatitis developing in the female mice. This, of course, is a C57 Black 6J issue with the female bias. And we also see about 16% of that 0.7% are sort of spontaneous mortalities that are found in the cage, which we don't always have the opportunity to do a full necropsy on. The situation's a little bit different in males in that mortality and dermatitis are the two major reasons why we lose mice in the 6 month to 1 year time point. We see mortality being still, spontaneous mortality being seen as a prevalent or a major factor in the loss rate in the slightly older ages. But again, this is only just 44% of the 0.14% of mice that we're losing on an average week from our aging colony. And then mortality is, again, a major factor as these mice get older and we start to see the emergence of more dermatitis. But overall, I think in large part because of the technician training, the enriched caged environment, and the high health status that the mice have in our aging colonies, and we don't see the prevalence of some of the other conditions that are seen sometimes in other aging studies like rectal prolapse, et cetera. We have more resources available on our website, and especially related to aged mice, but also related to other strains as well in the mouse phenome database. And that brings me to the end of my presentation today. I hope this has been a useful overview of some different enrichment benefits. So I'd like to thank you for your time today.
Okay, thanks so much. Thanks to ALAS again, and thank you, Andy. You did a great job, a lot of fun. And everybody take care, be safe.